this is FemmeSciComm's 6th vlog. Every Thursday, I post videos and written blogs about published women's health research studies on FemmeSciComm.com. I know that there are many women who suffer from reproductive disorders and want to keep informed about what research is going on to help them treat their disorders. For example, the best therapeutic options for women with endometriosis and uterine fibroids remain birth control and uterine surgery. But if these women want to get pregnant, these aren't really options. Research is going on every day to help pinpoint new therapeutic options for women who suffer from reproductive disorders. This week's post touches upon a reproductive disorder I've mentioned already, endometriosis. The authors of this paper that I'll be discussing look at DNA methylation, a concept I mentioned previously, as either a diagnostic technique or therapeutic target. So this paper was recently published in Clin Epigenetics, Genetic Regulation of Methylation in Human Endometrium and Blood and Gene Targets for Reproductive Diseases. The first author is Sally Mortlock and senior author is Grant W. Montgomery, both from the University of Queensland. So let's get into it. This study aims to find if there are DNA methylation differences between blood and endometrium at different stages across the menstrual cycle. DNA methylation is an important factor for gene regulation. Remember, I see it as a hoarder's home. Lots of boxes inhibit or stop you from reaching that person physically. Here, the boxes are DNA methylation, and high methylation means a specific activating protein, U, can't get to the gene, the hoarder, they want to turn on. Low or no methylation means the same activating protein, U, can get to the gene, the hoarder, they want to turn on. The researchers want to see which parts of the DNA are methylated in the endometrium and in blood. With this data, they can find out if these specific areas can be predictive of any reproductive disorders, or if they can gain any new insights on reproductive disorders. So we'll start with what they found. The researchers first compared DNA methylation profiles from blood and endometrium, and they found more sites of DNA methylation in blood. The researchers observed similar total methylation patterns in the endometrium between the different phases of the menstrual cycle. However, they detected significant changes in DNA methylation in the endometrium when comparing between the proliferative, right after menstruation, and secretory, right before menstruation, phases of the menstrual cycle. They then integrated their data with a public genome-wide association study, or GWAS dataset. A GWAS dataset comprises of thousands and thousands of patients and their phenotypes or characteristics. They determined 5 of their probes correspond with important genomic sites that are associated with endometriosis, a phenotype or characteristic. Upon more stringent analysis, they found that the genomic site corresponding near gene GREB1 associates with endometriosis risk. They determined that women's genotype or DNA sequence can vary between 3 types, CC, CT, and TT. Although expression is relatively similar between the different genotypes, methylation seems to look different. This GREB1 gene locus was already found to be associated with endometriosis risk, but the authors bioinformatically determined differential methylation near this site is also associated with endometriosis. So what does this all mean? This is the first study that looks at DNA methylation in both the endometrium and in blood across the menstrual cycle. As I've mentioned before, DNA methylation is an important factor when it comes to gene regulation. Because the endometrium is a biologically dynamic tissue, it makes sense that DNA methylation plays a role in the menstrual cycle, and that specific regions of the DNA are methylated differently in the endometrium versus in the blood. I mean, what if our blood shed every 28 days? Additionally, any change in normal DNA methylation can affect endometrial physiology, or the normal function in the endometrium. The authors found an area of DNA near the gene GRB1 that can be indicative of endometriosis when differentially methylated. I think this study aimed to introduce the use of DNA methylation as a diagnostic technique, which leads me to the caveats. Their patient cohort is small. The sample size comprised of 66 women of European ancestry. In addition, important menstrual differential DNA methylation was not found in blood, only through endometrial samples, which I've mentioned before makes sense. Also, remember, analysis of the results were through bioinformatics and meta-analysis. There were no functional experiments done. Overall, this study determined there are important menstrual differential DNA methylation in the endometrium, but this is not seen in blood. Although this study does not include functional experiments, the authors revealed a relationship of a handful of differentially methylated DNA regions with important genomic sites that may associate with endometriosis, one being GRB1. Finally, genetic or DNA testing cannot completely predict whether a person will develop a specific disease. I think the authors introduced that epigenetic testing may be possible in the future to supplement genetic testing. Thank you for reading, watching, and listening to my assessment on this paper. Keep informed, stay interested, and keep doing great women's health research. Thank you for watching. If you're interested in this content, please make sure to subscribe to my channel. Check out my website, FemmeSciComm.com, and you can follow me on Facebook, Instagram, Twitter, and Pinterest at FemmeSciComm. Thanks.